Welcome to the next in the Journal of Clinical Investigation series, Conversations with Giants in Medicine. I'm Ushma Neal. Today I have the distinct pleasure of speaking with Dr. Lori Glimcher, a world-class immunologist who helped define the transcription factors that make immune cells commit to being immune cells and to activate. Following her path of scientific discovery, she's also stumbled across key bone anabolic pathways, as well as become an expert on ER stress and lipoprotein production. Furthermore, recently she's turning her attention to tumor immunology. All the while, she's acted as an academic leader, serving as head of her department at the Harvard School of Public Health, most recently as dean of the Weill Cornell Medical School, and she's soon to take up the reins as president and CEO of the Dana-Farber Cancer Institute. Thank you so much for joining me today. I really look forward to learning a little bit more about your life.
Pleasure.
So can you start by telling me a little bit about your parents and what you were like as a kid?
Well, my dad was a physician scientist, which is an important thing because in many ways I followed what he did. My mom was a homemaker. But what was interesting about both my parents was that they were very early supporters of women. And there was never any question that my two sisters and I were going to have independent careers. That was never even doubted. It was assumed that we would pursue our passions, whatever they were, and that we would have independent careers. And I have two sisters. One of them is a year and a half older, and one of them is a year younger, so we've always been very close. And even today, they're really my closest friends. But neither of my sisters was particularly fond of hospitals or blood. So they never had any interest in accompanying my father to work, but I actually liked it, so I used to go in with him to work. He was chair of the Department of Orthopedic Surgery at Mass General, and then the chair of orthopedic surgery at Children's Hospital, and a biophysicist and a biochemist who studied bone, studied skeletal biology. So, in fact, he really created the orthopedic research laboratories at both of those institutions. So I would go in with him on weekends to the Mass General and sometimes go on rounds with him or just go into his laboratory. And then I worked in his laboratory as a high school student and as a college student as well.
So you think the biology and medicine imprint came on very early as a child?
I think it did, and I think it fit in well with my interest in taking care of people. I used to grow flowers, and then I would take a dead flower and try to see if I could revive it. On the other hand, I was a little bit callous, and I horrified my sisters one summer in Maine when I think I was about 6 years old, and I dissected a frog on the front step of our little rental cottage, and they were just horrified.
But it was a curiosity driven by figuring out what was inside?
Yes, I wanted to know what was inside of that frog. And my sisters, I have to say, are both tax lawyers now, so they had other interests.
So were you good in school?
I was always good in school. I was always impatient and quick to learn, but not necessarily the most tolerant or tactful person. I didn't tolerate fools gladly, and that was an aspect of my personality that I hope I have under better control now.
So what was your path towards medical school? You went to Radcliffe?
I went to Radcliffe, and from thence on to Harvard Medical School. I wasn't really entirely sure what I wanted to do in college. I did some acting. I took probably more literature courses than I did science courses. I can't say that I had a thrillingly wonderful GPA. In fact, I would never get into medical school nowadays. Absolutely never get into medical school nowadays. But I always liked thinking about the etiology of disease. That's what fascinated me, particularly once I got into medical school. So I got accepted to a number of medical schools, and I was going to take a year off because I started school a year early, so I was only 20, I had just turned 21 when I graduated. But my father said to me, you're probably never going to get into Harvard Medical School again, so this may not be the best time to take a year off. So I didn't. And when I arrived at medical school, my first reaction was, oh my God, I don't belong here. There are so many brilliant people in this class. And I would hear the other students kind of wandering around saying that they had graduated summa cum laude and that blah, blah, blah. And I thought, oh my God, I can't survive here. And I remember that first night, I picked up the phone, I called my mother and said, I can't do this. You have to come get me, which she did. She came and got me and she fed me some chicken soup and said, now you're going back and you're gonna be fine.
And you were fine.
And I was fine, yes, I was fine. And I loved it. I think, you know, from not being sure that that's what I wanted to do, to be a physician and or a scientist, it became very clear those first few months that I was in the right place.
So when during medical school did you start to think about immunology as your focus? Or did that come later?
No, that was an aha moment. You know, in the first year of medical school, you do various blocks. And one of those blocks in the spring was immunology. And when I started hearing about the immune system, that was it. I absolutely fell in love with the immune system. I was fascinated by it. I spent the summer reading about it with the help of Emil Unanue. I was also working, continuing my work in my dad's lab on the skeletal system. But I think that was just it. And then I started thinking about experiments that one could do in the immune system. And the thought that you could have such an elegant system that can recognize self from non-self. So I got very fascinated by autoimmune diseases at that point. And then of course there were the other 3 years of medical school. The 4th year of medical school I spent in Harvey Kanter's lab. And that was another, I think, eye-opening experience. I remember Harvey saying to me, well, I actually went around to see whose lab I could work in. And Harvey had just arrived as an assistant professor at the Dana-Farber. He had just discovered that lymphocytes came in more than one flavor. You had killer cells and helper cells. And when I came to him saying I was fascinated by lupus and by autoimmune diseases, he said, you have to understand the normal immune system before you can understand what goes awry.
So at this point, you know, you've spent summers in your father's laboratory, you spend your 4th year in Harvey Kanter's lab. So at this point, were you thinking, I want to be a physician scientist and run a lab, or were you thinking about doing a clinical trajectory?
I think I was already stung by the research bug. I did like clinical medicine, and I was good at clinical medicine. I wasn't sure how much of my time in future I would spend seeing patients, but you know, when you graduate medical school, you really are in a very primitive state. So, you know, my first night of being an intern at the Mass General, I got a page from the emergency room, and I will never, ever in my life forget the number of the MGH emergency room, 3326. I got a page, and my first patient was a cardiorespiratory arrest. And I thought, oh my God. But you know, down I went, and luckily, the senior resident at that time was Bill Seaman, which is how I first met Bill. He had just returned from the NIH, so he was a little rusty too. But together, we spent the night, took care of the patient, and you know, it's such a steep learning curve when you're an intern. And it's like learning how to be a car mechanic. I mean, you're acquiring a very complicated skill, which is taking care of deathly ill patients. So I loved my internship year. You know, you go from being somebody who really doesn't know how to take care of patients to being somebody who thinks they know how to take care of patients. But already by my residency year, I was getting a little restless and started again thinking about, you know, why does this person have scleroderma or lupus or amyloidosis fascinated me too. So I knew then that I didn't want to be a full-time clinician, and I wanted to pick a subspecialty that was limited enough that it would allow me to see patients It's a very small percentage of my time and spend most of my time in the laboratory.
So you decided to pursue rheumatology, right?
Yes.
But before that, you decided to go to the NIH to do some postdoctoral research?
Well, I had sort of decided both at the same time.
Okay.
And my first husband, who was a transplant surgeon, was also interested in training. So he went to David Sacks's lab, I went to Bill Paul's laboratory, and it was just a fantastic 3 years.
Well, you were there at a very heady time.
It was a heady time.
With Bill Paul and Ethan Shivak and Mark Davis cloning the TCR
and
right
Mark
next
Davis
to you.
came about the same time I did. He was a postdoc with Ron Schwartz, and Steve Hedrick was also a postdoc with Ron Schwartz. Mark Davis was working with Bill. It was a very heady time, and immunology was really premier at the NIH. So it was very exciting, and you know, it was a very productive time for me because I was mentored not only by Bill Paul, but also by Ethan and by Al Singer. I just worked with a lot of different senior investigators, and it was a really productive time. I had, in more ways than one, 2 months after I arrived, I gave birth to our daughter, Kayla, and I thought, well, I'll take some time off. And but before she was born, I had gone in to take the medical step, whatever, of the medical boards. And no, sorry, that was after she was born. So she was born, I was at home for 4 or 5 days, getting edgy. I took the medical boards, and I realized right then and there, no, I have to go back to work right now. And my mother was horrified. She said, "You have just had a C-section. You're supposed to stay home for 3 months." And I said, "I can't do it. I just can't do it." So I went back to work, 5 or 6 days after she was born. And that was just the right thing for me. I was getting anxious, I was getting depressed, and I realized that this was not good for anybody, including my daughter. So back I went to work, and then 2.5 years later, about 6 months before I left the NIH, gave birth to our first son. And I remember telling my mother, now, who was going to come down to Washington, D.C. to help, I said, "I just want to tell you that I'm going back to work the day I get out of the hospital. And if you have a problem with that, I just
want to prepare you ahead."
Prepare you ahead. So he was born, and she didn't say a word. She did not say a word. She stayed for a couple of weeks, and then she went back, and it worked out fine.
So what were you working on when you were at the NIH?
So I worked on a number of different projects at the NIH. Started out with something that was relevant to autoimmunity, and then segued into what I consider the major project, which was looking at the function of the MHC class II molecules. And at that time, they hadn't been cloned yet, of course. I was trying to produce a series of antigen-presenting cell lines that had mutations in their MHC class II molecules. And the scheme that Bill and I had dreamed up was to mutate a B-cell lymphoma using, I forget what we used, but some chemical, wait a minute, what did we killed all the cells. We used a chemical mutagenesis, then we killed off all the cells with one monoclonal antibody to class II, and then looked for cells that had survived that using another epitope. And I was told by everybody, this is never gonna work. This is not gonna work, but it did work. So that's where I learned, if you don't aim high, you are never gonna make major contributions to this field, and you've got to be a risk taker. We went back to Harvard because my husband had to finish out a surgical residency, and I was going to do a fellowship in rheumatology. But I just couldn't give up the laboratory because things were too exciting. So we went back. I had a 3-year-old and a 6-month-old. My husband was a surgical resident, so
he was not around.
He was not around, absolutely not around. And I was trying to be a full-time clinical rheumatology fellow and keep my lab going. So I think that those may have been the most difficult years of my life.
But at the time, you had an R01, so you're age 31. And you start a small lab.
Yeah, I did. I started a lab in a little corner, and I had an R1 because I decided to apply for an R1 when I was a postdoc, about the time that I was pregnant, about ready to deliver my older son. I thought, let me write an R01, which I did. It got funded, so they had to make me an instructor. So I came as a faculty member, even though I was a fellow. And I set up this little teeny lab with a couple of technicians to continue to work on these series of cell lines that I had generated that had MHC class II mutations. But I also decided this is the era of molecular biology. This, first MHC gene had been isolated. And I thought, I can't miss this revolution. And a couple of people said to me, no, you shouldn't try that. You've got too much on your plate already, so why don't you just be a cellular immunologist? But I said, no. You can't, this is the future. So I did an informal, unofficial mini sabbatical with John Seidman for about 6 months and learned the technology, and then hired a postdoctoral fellow who learned the technology, and we did that for a few years, but then I got kind of unexcited about structure function. It was fine. It was a good start, but I got fascinated by something I knew nothing about, which was how genes are regulated. And that's when I decided that we should figure out how MHC class II genes are controlled. That was the second phase of my career, where we isolated some factors that controlled MHC class II gene expression. We looked to see what the defects were in patients who had Behr lymphocyte syndrome. And that also was fun for a while, but then I got a little restless with that, and I got interested in how T cells differentiate. And that's probably what I'm best known for, is identifying the transcription factors that specify lineage commitment in T lymphocytes. And that again was, I remember the postdoc that came to my laboratory, Suzanne Szabo, and I said to her, how would you like to isolate the key transcription factor that controls type 1 immunity? Because we had already isolated CMAF using a yeast 2 hybrid screen. Again, that was a very new technology. But I always believed technology is just technology, and so you need to just jump on board. And we did that. We did that actually when we isolated transcription factors that we thought controlled MHC class II gene expression. We were wrong about that, but that was just when Phil Sharp's group had published their work on doing southwestern blots. So we did that, and we pulled up what turned out to be XBP1 and others. And so the next step was to do a yeast 2 hybrid system, which we did, and that yielded us CMAF, which controls interleukin-4. And after that, when Suzanne came to the laboratory, I said, well, we really know nothing about type 1, Th1 cells, which
everyone had been focusing on type 2.
Everyone focusing on type 2 and interleukin-4, and we had done a lot of promoter bashing in the interleukin-4 promoter, as had others, and there had been a lot of promoter bashing for IL-2, which was at that point thought to be a Th1 specific cytokine, which it turned out not to be. And Jerry Crabtree's lab had identified the NFAT family. So we really, nothing was known about interferon gamma, so we couldn't use that as a promoter. We decided to do a yeast 1 hybrid screen, which again everybody thought was crazy because that had been done with multimerized sequences, and I thought that we really needed to do it with the whole IL-2 promoter. And Suzanne thought, well, let me try to first do it by expression cloning. So she tried that, and that didn't work. So she said, okay, I'll try the yeast 1 hybrid. And we piloted it by using CMAF as a proof of principle, putting in the IL-4 promoter, and then seeing if we could pick up CMAF, and we did. So off she went, and we picked up this factor, which we named T-box expressed in T cells because it was a T-box binding protein. And it took us a while to figure out what it did, because everybody at that point was doing transient transfections. And this damn protein did not transactivate the IL-2 gene. It repressed it. And why? Why did it repress it? So just at that point, the technology to do retroviral gene transduction of primary cells emerged. And I said to Suzanne, that's what we have to do. And she came running into the office a couple of weeks later, and she said, you're not going to believe this, but I've transduced primary T cells and they're pouring out interferon gamma. And
so that was your moment for champagne.
That was, I said, I can't, I didn't believe it at first. She had to repeat it a few times before I believed it. And I realized, of course, that the yeast-1 hybrid system, all you're doing is forcing something to attach, but it doesn't have to be. And you have a false transactivator sequence, right? Transactivator domain. So that it was binding to the IL-2 promoter, it certainly was, but it was repressing it. And that made sense, because as naive progenitor T helper cells differentiate, they lose the production of IL-2. And TBAT was a factor that was pushing that differentiation. And then it turned out that TBAT was better than we thought, and that it had really key functions in almost every immune cell, both in the adaptive and innate immune system.
Well, talk to me a little bit about when you first came across Shinuri-3 and how perhaps your father was helpful in exploring that.
He was helpful at that. So that was, again, there've been a lot of serendipitous events. And I guess you can choose to ignore a phenotype or to pursue it. And sometimes you have to ignore it because it defocuses you, and it's, I think, very important to produce a body of work that can really contribute to the field. So you need to have a central focus, but maybe I'm just easily distracted. But so we were actually looking for TBAT. And a postdoc did a subtraction experiment, subtracting Th2 cells from Th1 cells, looking for the factor that was critical in Th1 cells. And what he pulled up was this protein that was then called KRC, been isolated by another group and thought to control VDJ recombination in the thymus, although those were all in vitro experiments. But it was really expressed heavily in Th1 cells, so we thought maybe that's what it does. So we knocked it out and could not find an immune system phenotype. We looked everywhere. We found a modest effect on the production of interleukin-2, a transient effect on the production of interleukin-2, because it seemed to be a co-activator of AP1 family members. And I had, at that time, a postdoc and a graduate, MD PhD graduate student in the lab, and we sat there. I said, there has to be a phenotype in the immune system. Have you looked at every single organ? I mean, you've looked at the thymus and lymph nodes and bone marrow, and the graduate student said, actually I haven't really looked at bone marrow because I'm having trouble extracting the marrow from bones. I said, really? Why is that? He said, well, they seem to be occluded. And I said, why don't the two of you walk down the street to Children's Hospital, where my dad's lab is, and take an x-ray of the mice? And they looked at each other like, okay, let's just humor her because she's always coming up with these crazy ideas, and most of them don't work. They went down the street, took an x-ray of the mice, and came running back because they had unbelievably high bone mass, and the bones looked normal. So you can create high bone mass with fluoride or other things, but that bone is biochemically and biophysically not good bone. It's not lamellar bone, it's woven bone, or it's brittle bone. This bone looked good on x-ray, and it did not involve dysmorphogenesis. So again, at that point, most of the factors that controlled skeletal biology were factors that were hereditary, that caused craniofacial abnormalities or cleidocranial dysplasia. So the focus had been on developmental anomalies in the skeletal system, not on adult bone mass. And this factor didn't do that. It worked postnatally. Well, I mean, I had skeletal biology in my genes. I had worked in my dad's lab as a college student and as a high school student, and he was just down the street. And so we set up this collaboration. I knew nothing about bone, other than the fact that I was a rheumatologist, so it was somewhat relevant, bones and joints. And we began to explore how this factor worked, what it did, and how could we harness it for therapeutic purposes. And we are still working on SHNURI-3. Along the way, we formed a wonderful partnership with Merck, and that partnership allowed us to make a lot of progress in understanding SHNURI-3, but also allowed us to look in an unbiased fashion for other factors, other genes, proteins that control bone mass, and we found several of those as well.
Not only are you this world-class immunologist and biologist and physician scientist, all the while you have taken on these massive academic leadership positions. So first as chair of the Division of Biological Sciences at the Harvard School of Public Health, and most recently as Dean of the Weill Cornell Medical School. So have these administrative responsibilities taken you away from the science? Probably necessarily had to a little bit, but what have you learned from taking on those roles? And why did you take on those roles?
So first of all, I have to say that being the director of the immunology program at Harvard and being the director of the Division of Biologic Sciences at the Harvard School of Public Health were not huge administrative jobs. They were administrative jobs, but they weren't huge. And like any other faculty member, I served on multiple search committees and did multiple other tasks, like being on the Larry Summers Task Force and so forth. So going from spending 95% of my time in the laboratory at Harvard to being Dean of Weill Cornell Medicine was probably the steepest learning curve I've ever had. Luckily, I'm a very well-organized multitasker, but I really hadn't tested out that, other than raising 3 kids and having a lab, hadn't really tested out whether I could also continue research and run this big institution. I was a very, very unconventional choice for dean. They really took a chance on me. Almost all deans have had previous clinical positions. They've been chair of Department of Medicine or previous deans. I never had any interest in being chair of a Department of Medicine. I had numerous calls, but this just never appealed to me at all. But this, when the search committee called me from Weill Cornell, I thought, I think I was 58 at the time, and I thought, look, I want to have a second career. I want to do something that goes beyond my own lab, because I think I can have a bigger impact in academic medicine if I focus on other people. And that was really why I wanted to do it. So I spent the first, I would say, year really focusing on the research enterprise. And then I realized that our medical school curriculum hadn't been altered since 1996 and that we had better get going on that. And so we assembled a committee of faculty and came up with a really interesting new curriculum, which the students seemed to love most of the time. And then after that, I thought, well, I think the physicians' organization could be even better than it is. It's not broken for sure, but we can always make something better, and so we turned our attention to that. None of this did I have to do alone. I had such a wonderful group of senior management. I had such a wonderful senior management team that were so helpful, and when I first got there, Carl Nathan was just essential to me. One of my heroes.
Mine too. Now, you mentioned something when you were speaking. You served on the Larry Summers Task Force for Women in Science and Engineering.
Did, yeah.
What was that experience like? And do you think that it had a positive impact? And where do things stand right now, both at Harvard and elsewhere, for women in science?
I would say that I'm disappointed at the rate of progress for women in science and medicine. If you had asked me 30 years ago, where would women be now in science and medicine, I would have said, well, we'll be right up there. We'll be in senior leadership positions. We'll be running places. we're gonna, we're gonna reach equality with men. That has not happened. There are still very few women in senior leadership roles. And that's true at all institutions, including Harvard and Weill Cornell and anywhere else, right? We just haven't done well enough. And one of the things that I've always cared a lot about is mentoring, and mentoring both men and women, of course. But I have a special place in my heart for women because I know how hard it is. I know how hard it is to raise a family, have a spouse who's incredibly busy, and pay enough attention to your kids. And I don't want any woman to say to herself, well, I really would like to have kids, but I'm not going to do that because of my career. Women shouldn't have to make that sacrifice. If you don't want children, fine. Okay, your life will be easier. But if you do, then, have them, for God's sakes. It was never a question with me.
I've often re-quoted you saying, I've got a brain and a uterus, and I use them both.
Now, that's not my quote,
No?
That's Patricia Schroeder.
Okay.
It's Patricia Schroeder, the congresswoman from Colorado.
Well, it's still an excellent quote.
It is an excellent quote. I love it. I use it all the time. And she certainly did both. I think you have to put your money where your mouth is when it comes to women, right? You need support, and I've spent a lot of time during my career mentoring women. And you need intellectual support, as you do for any mentee you have. You also need to have emotional support, because women really tend to be less self-confident than men. And that includes all of us.
So at the end of a particularly complicated day, and you are home and you're thinking, why did I do this? Why did I make this choice? And you had to do it all over again. What is the alternate career that you fantasize about if it's not a scientist or a doctor?
There isn't one. I love the career I've chosen. I can't imagine, honestly, doing anything else that would bring me as much joy as doing this.
Thank you so much for joining me today, Lori. I've really enjoyed speaking with you.
Thank you, Ushma. It was great.